Okay. Hello to everyone who is here. Today we are going to have a small workshop on how to use the LivingNorwayR package to both create and work with Darwin Core compliant data archives. So if you're watching the recording and you haven't watched the introductory talk on Darwin Core from last month, I recommend you do this first. But otherwise, we will jump in now. So the way this is going to work is that we're going to have an introduction to the R package with some live coding by Matt Granger. And that will be recorded and available afterwards. And then we will have an interactive session where we will be working on some different data sets on making them Darwin Core compliant and fiddling around with them a little bit. So that's it for introduction. There's not much more to say, but I will hand over to Matt now.
Hello. So I'm going to try and do some. Well, I cheated a bit on the live coding, so but I'll try and do some bit later. But I'll just go run through the LivingNorwayR package. I've got some slides to share. Of course, I can't see them now. So I'm just going to turn my camera off. Right, can you see my slides? Yes, getting thumbs up from the corner here. Okay, so the LivingNorwayR package is a package we developed to interact with the LivingNorway Norway project and the LivingNorway data portal. The idea is that we can provide a workflow for creating a Darwin Store standard compliant data archive, a data package, and this will facilitate fair data and metadata sharing. So there's two things we can do. We can get data from the portal. So the data portal contains examples of data sets from Norway at the moment, but you could also use the same approach for getting data from GBIF. But I'm just going to show you from the LivingNorway portal, and you can see that on that page. So this is just an example of one of the data sets. When you go to that the portal, I'm sure Chloe can give us a link to the portal in the chat. You can see there's a thing called the key. If you copy the key, you can then use that to download the data in the right format for the LivingNorwayR package. So this function is getLNPortalData. So using the LivingNorwayR package, we can then call down that data using that function. And what it does, it brings down a object, which is a class DWC archive, a Darwin Core archive, and it's an R6 class in R. So in R, you have different class types. So most of you are probably aware of different classes like a list or a list or a data frame or something like that. Here we've actually developed our own class, and that class is a Darwin Core archive class. And that class has functions that run on it. So you'll see in some of the functions in the code that actually there's a little bit of difference in how you express the code when you're writing R, but it's not a huge difference, so it's not a massive barrier. As we've spoken about a few times before, a Darwin Core archive consists of a core. So this could be a table of data about your sampling event or about your species that you've species occurrences that you've recorded. It has extensions. So these could be measurements about the species that you've recorded if your core is an occurrence dataset. It has metadata, which is the descriptive data about what the data is about. It's useful for other people to find out. And a thing called a metafile. Now, the metafile is a thing that tells how these different files, the core, the extensions, and metadata, are related to one another. So it's a useful, it's basically a package, a zip file of these different things. So using the LivingNorwayR package, you can see here that we've downloaded our Beetle data, and then we put the $sign next to our data and get this these functions. So this is the difference I was talking about with the R6 classes. And you can bring out the core table, the extension tables, all the metadata. So as an example, bringing out the core table. So in this case, it's an event core. So you'll see that when we bring out the core table, that you get a class of a GBIF event, which is a DWC generic class of an R6. So the event is data about a sampling event that's happened, in this case on beetles in Norway. The core table you can export as a data frame. So again, these functions, you can, once you have the object called core table, you can do $sign and then have a number of functions. So one of them is to make a normal data frame. So here I've made a data frame called core table DF. You can see you can use it exactly the same as you would use any other data frame. It has some simple functions like names and those sort of things. But we can also use this data here to make visualizations. So let's have a look at this one. So we can make a map. We can find the decimal longitude, decimal latitude of these sampling events, and then we can plot it into a leaflet map. And these are in Norway, and these are the sampling events. In this case, there's beetles in areas where there's been, I think it was electricity pylons, and this is just the map of those. And because this is another thing you can do is get the extension tables. So here again, remember our object was called Beetle data, and now we can use our $sign to get a function. In this case, it's the extension table, and the extension table is actually an occurrence data set. So it's the species that have been recorded in each of these sites, and then we can export that as a data frame. You'll see here I'm using an index of 1 for the occurrence extension. That's because there's two extensions. There's a current extension and another extension there. So I'm just calling the first one. And again, this becomes a data frame. So you have exactly the same thing. You've got a data frame that you can use as you would do normally in R. So you can run some code. Maybe you find an image on R file pick and find an image of the species that you're measuring. So here's just one of the species of in the data set, species of beetle. Sorry, there's a genus of beetle and the different species. And here is one of those individual images that you can have. So you can use it just as exactly the same as you would do the data frame. And finally, we can also get the metadata. So the metadata from the object. So here, metadata is again from our Beetle objects, Beetle data object, and the function is get metadata. And from that, we can get our title and creator, so who made the data set. And then you can put that into into R, you can write it into Markdown, all those sort of things. So you can do here, I've just pasted together. The project is its title, and it's created by the creator. So their first name and the second name. And that prints out what the title was and who it was created by. So that's bringing your own data down from LivingNorway. You can do the same thing from GBIF. Thank you. My brain went blank then. From GBIF, using that key area. But we've designed this function within the LivingNorwayR package to bring down data from the LivingNorway portal, data portal. Okay, so this is the more in-depth part. So how you can convert your data to LivingNorway format. And some of you have got some data to try this on later on today. I'm going to try and avoid laboring the point too much, but I'll give you an example of how you can do this and what sort of functions there are. So first is thinking about what a data set is, and an ecological data set. It generally has very similar elements. And if you've been listening to a few of the other talks in this series of seminars, you'll begin to know and recognize some of the different elements of this sort of Darwin core approach. You can have most data in ecology is similar, although I think many ecologists probably might not say that. They'll say that all our data is very different, but that's you can actually broadly put them together into one group. So you might have an event, so some sort of sampling method. So if you've done a line transect or camera trapping at a certain place at a certain time, you have an event. So all the information about that is the event data, and that can go into a separate table. You can have a set of occurrences, so all the species that you observed at that site during that event would all go into a list of the species you recorded. And often you might have a measurement or fact table as well, so the things you've measured. So it could be, perhaps you've caught some rodents and you've measured their tail length or their weight or something like that. So that all that information go in. And then you have metadata, and the metadata is probably the most important part. It's the data about the data. So it really describes what the data is about. So it makes it easier for people to find your data and make use of it as well. So these are the sort of structural elements that you can put in. In the Darwin core archive approach, what basically happens is you have separate tables into one zipped folder, big folder of these different things. And obviously we'd never really, as ecologists, go out and collect data in this way. We don't separate our tables as events and occurrences and measurement facts. We often have it in columns across. So you need to do a little bit of reshaping to get your data in the right format for Darwin Core Archive. And the LivingNorwayR package is designed to help you develop that sort of reshaping of data and then providing a LivingNorwayR Darwin Core Archive file, which is a data package together. So we've got these elements of event, occurrence, and measurement. In fact, there are other extensions you can put in, and we've got a whole list that we can look at. But for the moment, these are the sort of the core ones we talk about. So let's use an example, and we're going to use the well-known Palmer penguins dataset. It's a dataset that's openly available on R. It's available in R, and it's a really useful dataset because it's good for sort of ecological approaches, and it's a bit more politically correct than the IRIS dataset, which was trying to replace. So we have a dataset of observations and measurements of 3 different penguin species in Antarctica: Chinstraps penguins, Gentoo, and Adelie penguins. So you can pull in the data using the Palmer penguins package and have a look at it. And what it consists of is a study name, a sample, the species, the region, an island. The stage is an adult or chick. The capture number, so the individual that was caught, had some breeding that happened. Did it lay an egg? Then they took some measurements about the penguin itself, its sex, and they also took some blood samples as well. So this might be sort of the typical type of ecological data one might collect. So here's an example of how they measure it. So the bill length and the bill depth it measures. So it just gives you some information about how things were measured, what they measured, and from each different individual's penguin. So we've got our data, we've gone out, we've measured our penguins on these different islands in Antarctica. We've come home, we're having a hot drink because it's been cold. We want to now convert our data to a Darwin Core compliant data archive. So the biggest thing, and it's something that I find difficult, is deciding on how to structure our Darwin Core archive, because we can have the core file could be the event, but there might be cases where you just have an occurrence data set that has that as the core, and I think it's something that's quite sometimes a bit individual about how you do that, but we can think about what the core classes are. So LivingNorwayR package gives you a function to actually call in all the different core classes, and there's quite a few that are available. So you could look at those and run down the list of different core classes. There's a couple here. You can have a look at that. Look at the definition. So here we've got a GBIF event core. So that's a class, and it's an action that occurs at some location during some time. The examples are things like camera trap capture, marine trawl, some sort of process that leads to collection. And then the other core you can have is a GBIF occurrence. Another example of the core you can have is a GBIF occurrence, which is the existence of an organism at a particular place at a particular time. Examples here are a wolf pack on the shore of a lake, a virus in a plant leaf, a fungus in Central Park, all at certain dates and certain times. I won't go into the full list of these different things. You can have a look at yourself. So if you call from LivingNorwayR get GBIF core classes, it will tell you the different core classes that are valid. So in our case, we've got this measurements of penguins on different islands. So we can actually look at these as an event, and we may put the as an event core. And we have species data as well, so we can actually put the occurrence data as an extension. So we have that in a separate table, but it's still all linked back to the event. So different extension classes, there's quite a few of these as well. And you can have a look at that list by doing get GBIF extension classes. And there's lots of different examples. So this first one is multimedia. So you might have some sort of video or sound recordings that go with your captures. It could be all kinds of different things that you can actually have as an extension course. And one of those is an occurrence. So there's quite a few different choices, and that gets a bit overwhelming. And I think that's something that we need to develop more nuanced guidance about how to choose which is your core and which are your extensions, and what to put in those things. But we can talk about that another time. So for the event core, we can get a list of the terms associated with an event using, again, LivingNorwayR to get GBIF event members. Here we've got an exhaustive list of all the event members, so all the things that are valid with GBIF as event members. And these are all the terms that can go into an event. And you'll see some of them, like this one, are no longer used, but they're still left there to sort of ensure that there's compatibility to the past. There's all kinds of different things you can have, different things, different standardized terms that you can use. So these terms are things like the event ID, so the event date, the sampling protocol, so how the sampling took place, sample size, the units. You might also have other things like a parent ID, so you have events that are nested, hierarchical, and put them in. Country code, so where it was, the location, decimal latitude, longitude, all kinds of things like that can come in. And we're going to go through a few of those and just show you, what it means in relation to the penguins dataset. So these parent events are this hierarchical structure of sampling, which is probably quite common. You might have different sites that you go to, different types of even sampling that might come into it. So you can have different structure in your events. So in this case, for the penguin example, each study name is a different exhibition. So where they went out to measure penguins, and that could be a separate parent event for each event, so each sampling event. So parent event ID has to be unique, and we can use the UUID package in R to give us generate some unique IDs. And in this case, we use the penguin data, and we have just using tidyverse deployer functions to sort of tidy up and reshape our data set. So the penguin data, we can have a column called parent ID, and it generates an ID for each one. And then we can add each date in as an event date. In the penguins data, there isn't very clear about what the events are, but they have these egg laying dates. So I've used those as a proxy for the event date. We can add some scale, so the continent, the island group. So these are all the terms. So everything in bold in my slides are the terms that are standardized and acceptable within GBIF in this case. But it's also the same thing for LivingNorway that we use. And we can just go through using tidyverse again to run through the code and to make each column, or make a new column for each type of term that's in there. So for this case, we've got continent, which is Antarctica. So that's all the same for this data for penguins. And the island group is the Palmer island group. So that's our parent event. So that's upper level. And then we go down to our event, which is the sampling. And again, there's another set of terms that are acceptable in the events. And we use those to identify, to tag each part of our data set. So again, we've got things like the owner. So whose data is it? And this has a code. The type of data, so this is an event data, so we call it an event. And the name of the data set, the Palmer penguins. Again, each event needs a unique ID, and you'll get sick of this soon, but there's each thing needs a new unique ID. So parent ID is unique, and then event ID is also unique. And as you go down the hierarchy, you have different IDs that are unique. Okay. Again, we can have the date, the event date for each of the events. And again, I just use the date that the egg was laid as a proxy for the event date. Sample size and sample size unit, which are standardized. So we can add these columns in. The sampling protocol. So this is useful for to understand what has been done during the sampling and how it was done. And you can get the sampling protocol for the Palmer penguins from if you search in the R console. And all the parent events have the same protocol. So here you can see that the protocol is just plain text here. So each season study nest where pairs of adults were present were individually marked and chosen before the onset of egg laying, okay, and so on. So you can actually paste in that text as your sampling protocol. Country code and country, Antarctica AQ. The location it can be a unique identifier, again, using the UUID package, or it can be specific to the dataset. We know what the region is here, so we have different islands, and we can paste that together to make a location ID. So the region is Antarctica and the island name of each island that were visited. Decimal latitude, decimal longitude, probably the most important things for some of these data is to have this. And we can add that in using, for each island, we know we can work out where they are and add that in. This geodetic datum, in this case, it was WGS84, but that's the default that goes in. So you don't have to actually put that in. That gets attached. If you upload your data to GBIF, for example, it automatically assumes that it's WGS84 unless you change that beforehand. Some sort of coordinate uncertainty, I can add that in as well. So all these things coming together. I'm going to flip through a bit, but then what we do in the LivingNorwayR package is to then develop a data frame containing all those elements that we've just gone through and some more that I haven't talked about. So dataset name, the owner, parent ID, event ID, and so on. And we wrap that into a data frame, join it together with the parent event data frame to make a final data frame. And we end up with this GBIF event. So this is back again using the LivingNorwayR package. We're using this initialized GBIF event. We need the data frame. We have this ID column information, and in this case, it's the event ID, which is the column that it needs to know. And this function here is name automap equals true. So what you could do is, rather than going through all the stages I've just shown you now, you could actually do this manually. So and go through and add a list here. So name automap equals false, and add a list of the names for each of the columns, and link them up to a recognized standardized name within the event. So you could do that. It just takes you probably a lot longer, and you need to know all the different names of the standardized vocab for the events. So if you can go through the process I've shown you here for the penguins, in the same way of identifying those column names with a term that's accepted by GBIF, for example, then it's probably a lot easier for you to do. The occurrence extension. So this is the data on the species that you've recorded, on the individuals you've recorded. So where the occurrences were. So it's in our case, it's the different penguins, individual penguins. Again, using the LivingNorwayR package, you can get the occurrence members. So what things are valid for an occurrence. And you'll see also again that you have lots of things that are no longer used. So keep an eye out for those. But there's a long list of different things that are valid terms within an occurrence data set. The list goes on and on. Lots of them. There are recommended terms for GBIF. These are the occurrence ID, so again, the unique ID, how the record was made, scientific name, the event date, so that's also included in the event core. And there's some recommended terms that you might want to put in, where it was, the species, the taxonomic rank, and how many there were, those sort of things can all go in. So for our example, this Palmer penguins, the question code can be Palmer Station, Antarctica, the long-term ecological records. And the occurrence ID, again, has to be globally unique. We can go through this process. Again, I'm not going to run through all of these because it's just me talking and showing you code. It's probably not very interesting, so you guys have it very soon, but I'll flip through very slowly. Each time you are making a new column with the standardized term and adding that to your data frame. Importantly, the scientific name and vernacular name we can put in. So in our case, we can do that using the data available in the Palmer penguins. So you have a scientific name that needs to be standardized, and it needs to be needs to match to a known set of scientific names. But that's all available on GBIF as well. So you can do that. And so on, keep adding these classes. And finally, again, as we do with the event core, we can put it all into one data frame here. So we're taking all the different type, collection code, basis record, and so on. And then initializing a GBIF valid occurrence table. Okay. So that's the data frame that we're using is occurrence extension. The ID column is the occurrence ID. And again, we're using this name automap because all these terms up here, the family, order, class, file, etc., are all acceptable terms for the occurrence table. Okay. The final extension that we're going to talk about today is the measurement of facts extension. So that's where you've gone and you've measured something about, in our case, penguins. You've measured their beak length and their beak depth. You've looked at their blood, for example. So you can actually, again, have a look at this. All the classes that are available or valid for this extension, which is the measurement of fact extension. There's a couple others you might want to use, which are also available through the package. So there's extended measurement of fact, which is one that maybe you might want to use. And there's some, we had some discussion about whether using this data we should be using an extended or a measurement of fact. So there is some debate that still needs to be had about what's the best way to do things. Again, we have these standardized things and a list of properties that perhaps you should put in if you're using, you want to be GBIF compliant. The GBIF standards are very wide, and so there's quite a few different things that they suggest you might put in, but there's very little that's required. So it's good to be free and open, but it sometimes leaves you a bit lost about which ones to choose. So these are some of the things you might want to put in: the measurement type, the value, the unit, and all those sort of things. Okay. So for example, we could use measurement ID. Again, we need this unique identifier. So each individual measurement needs an ID. And we can include lots of these measurements. So from our data, we've got the length of the culmen, the depth of the culmen, so that's the beak, how much of these different elements within the blood, the body mass, the flipper length, and so on. And we can all include those all in our measurement of fact table. And what we need to do is reshape the data again to make this pivot it so that each measurement type, so the culmen length, the culmen depth, and so on, are all going to one column for the measurement value. So we just have one column with each different measurement value in, and that way it gives you a very long table. But because you're doing it within this process, it won't affect how you do things. It's just a different way of thinking about it, really, format it. And then finally, we can create our measurement table with this initialized GBIF measurement of fact table. Okay, measurement of fact, sorry. And again, exactly the same parts of the function: the data frame, the ID column, measurement ID, and name automatically equals true. Now, the most important part, which is something that we often think about, is the method. Well, something I often think about, but something that people don't often think about themselves is the metadata. So it's the data about the data, which is very important. It's an important part of making these data archives. So it helps to make it more shareable and more available for other people. The standard for Darwin Core archives is to have this metadata in EML, which is ecological metadata language, which is a type of XML, which is related to HML, so different computer languages, but it's designed specifically for handling ecological metadata. There's a few different options in R of how to write metadata using EML, but one thing is they are all of them are really hard and tricky, and there's some weirdness that goes on. So it's something that's probably tripped us up the most in this whole developing this package is this metadata development, and we're not quite there yet, but we're getting close. So our vision is to have an R Markdown script that you can add in. You can render to a data report, so you can write about your data, the metadata of your data. So where it was done, who did it, maybe an abstract, how it was done, the sampling protocol, that sort of thing. But also when you knit that, you could end up with EML at the same time, so valid EML at the same time. The current version is a little bit buggy. Well, a little bit means a lot sometimes, depending on the EML, but there's a lot of development going into that now, and we should have the version released by the end of this year that actually solves a lot of the issues. Yeah, just to tell you what that's about, what we're doing is actually taking the whole EML standard, which is massive, and actually using that to base our functions on, which means that we can do everything that you could possibly ever want to do with EML, but it's a big thing to do. So it's a lot of work we need to put in to make sure that we can do that. We're close. We just need to do a few more tests, and hopefully by November we'll have a big testing round, and then by the end of the year, the full package can be released with this EML thing. But I'll give you an example of what sort of things it can do. So we can write in an R Markdown file. So I'm assuming everyone knows what an R Markdown file is. It's basically a file that allows you to write both code and text. So you can have code blocks. So here at the top of here, you can see the code block. Here's library LivingNorwayR. And then you can write some plain text. But you can also embed your R code within that plain text. And the idea is that you can see that we can write some text about our data, and in this case, it's text about how the data set is, how the metadata is tagged. And then we can have these code blocks, or these code snippets. And each tag is an element of the EML. So something like the title would be the Palmer Archipelago Antarctica penguin data. And we can put that in as a tag that's hidden. So when we render this to HTML, all these tags won't be shown, but the outputs of them will. So here's the outputs, and you can see I've forgotten to do echo equals false on my top one there, so the library showing. But you can see that we've got some of the plain text, and then you'll see that there's also bits where the code has rendered to the output rather than showing you the tag. And our thinking, what we hope will work, is that eventually one day you'll be able to write the full report in Markdown about your data set, and then just run our function, which will then pull up all the tags and then order them correctly for the EML, and then render you both HTML, so a nice, pretty looking HTML document, and then your EML as well, which is hidden underneath. So that function, we've written the function. It does occasionally work, but there's also lots of bugs with it. So give it a go if you want to. It'd be useful for us to see where it falls down some more. But yeah, just to be warned that it does. It breaks down half the time at least. What you can do there though is that you've got metadata, initialize metadata. So metadata is a type of object in our package, and then you just point to where your RMD file is. So in my case, my RMD file is in my workshops folder here. And what our function does is then takes the RMD file, and it does two things. So it renders it first into HTML, and then it pulls out all those tags that we saw before. And the idea is that you end up with a valid EML file for sharing. So that's where it falls down a bit with the making valid EML. It does make some EML, but there's some issues, and that's why we need to go back and get the whole schema before we do it. So the final step of this whole process would be to bring together your different events, your different extensions, so your different core files, your extension files, and your metadata all into one file, this Darwin core archive. So in our case here, we would call our core Darwin core table is our event table, which we've called GBIF event. Our extension tables, we've got two of them. We've got an occurrence table and we've got a measurement or fact table. And you can link these together using C, so the same as you would do for any other R code. there. And then our metadata. And our metadata is what I just showed you, that RMD file that we just turned into an EML file. And we can initialize a Darwin core archive using our core, our extensions, and our metadata. And what that does is it produces a fundamentally a zip file that can then be used, uploaded to things like Living Norway Data Portal or GBIF. At the moment, there's still a couple of steps you need to do. You need to go onto the IPT, so that if you've got an integrated publishing toolkit within your institution, and upload that file, the Darwin core archive file. But we're looking to, in a next stage of development, is to actually run from R straight up to GBIF for Living Norway, depending which way we go. But something that you can just run a button and it will do. So we're not quite there yet, but we're getting there. So I've spoken to you probably enough. So we can actually play with the package, see how it works, see where the bugs are. But if you need some help with the package when we're not in this session, go to GitHub. I'm going to add a link to my slides and the GitHub in the chat once I finish talking. But go to GitHub and you can submit an issue, and you can just link onto these. If you want to contribute, so if you want to add some code, or you want to add use cases or some help us with our documentation, because our documentation is pretty lacking a bit, please just go have a look at the contribution guidelines and do a pull request through GitHub, or add an issue if you don't know how to do a pull request. And then that'll be really useful for us to develop this. So it's not just something that we're developing by ourselves, it's something that's wider. So yeah, there are guidelines for contributing and how you do that. So please have a look if you want to. And I think that's me done talking. I'm gonna have a breather.
Okay, so thanks so much, Matt. I feel like now we can actually just start playing with the package a little bit. That was a really nice introduction, and always good to actually see it linked see it linked to a sort of data that looks familiar-ish, at least. So at this stage, we're going to stop recording, and then I think we'll maybe take a short break before we start with the practical part. So I'll stop the recording, and for everybody who wants to stay on for the hands-on session, let's be back at 2:55.